So for any specimen that is received into the lab, of course, it should be labeled. And so when collecting a specimen, you should always make sure that you properly identify the patient, or the phlebotomist should properly identify the patient before collecting the sample. If it's something like a wound culture or any other tissue culture or anything, then the physician may be the one to collect that. It should be labeled, and of course it should be labeled at bedside, at the patient's side. And it should contain all of the necessary information, as well as match the order that comes with it. So whether it's a paper requisition or an order in the computer, the information on the container, the collection container, should match that order and requisition. So for example, if the patient's name is spelled one particular way on the requisition, then it should also be spelled the same on the patient's sample. And sometimes you can have issues where a patient may go by a nickname, and so it may be written on the sample with the patient's nickname versus the given name. So those are things that need to be double checked by the person that is collecting and labeling the sample. And so on the sample itself, the sample is typically labeled with at least 2 identifiers, and it depends on the protocol for the facility as far as what they will accept. But typically something like the name and date of birth, or the name and hospital identification number or medical record number. So there should be 2 unique identifiers to that patient on the sample itself. However, most places have gone towards barcoded labels, so most of that information will print on it rather than being handwritten on that specimen. And so things that will come on the requisition will be things like the patient's name, the identification number, the patient's date of birth. Sometimes it will also include the age, the sex of the patient, the collection date and time, the ordering physician, and where the specimen source is. So for example, a culture, if it just comes on a swab, you may not necessarily know where it comes from just because it's on the swab. So it could have been a throat culture, it could have been a nasal culture, it could have been a wound culture. So the specimen source is important so that you know what organisms you're potentially looking for as pathogens, and to know which culture media needs to be set up for those particular pathogens. The way specimens are collected is very important. You want to make sure that you're actually collecting during the acute phase of the illness, so this is when it's kind of rapidly onset. You also need to make sure that you're minimizing any sort of contamination from the normal flora. So with a throat culture, you just want to get the tonsil area, not necessarily the entire oral cavity. So you want to make sure that you stick to just the area where you're looking for the infectious organism. You should have collection material available to those that are collecting it, as well as specific instructions for the particular type of collection that is being performed. And so in some cases, the patient may be the person who's collecting the specimen, for example with a stool specimen, so those instructions should be very clearly written for those patients. And the collection should be completed prior to any sort of treatment, so before antibiotics are given, or before any sort of antiviral or antifungal medications are started. Specimens should be transported to the lab usually within about 2 hours, just to make sure you can maintain the viability of that sample. However, if it is delayed, then there are different preservation techniques that can be used to help ensure that those organisms stay viable. And also the containers should be leak proof, and they should be transported in a biohazard bag, because you never know what sort of specimen as far as the pathogen in the specimen. So it could be something that could cross-contaminate something else, or it could leak out and be a hazard to those that are handling those bags. So you always want to make sure that you're avoiding any sort of contamination. Some of the different specimen preservations that are used for micro include boric acid for urine samples, and so you can see the yellow tube in the middle up under the ONP pack. So you have a yellow tube, it's a BD vacutainer, and so what that does is it has a little bit of the boric acid in it, and the purpose of the boric acid is to maintain the colony count so that those organisms don't overgrow before it has the opportunity to be cultured. With feces, you get it's a buffered formalin that is in those packs, so you can see the parapack with that orange top at the top. And so that's an example of a collection container that may be sent home with the patient to collect. Then you have different holding medias like Stewart's and Amy's media. And so you can see some swabs in the lower right hand corner, which are used for transport of some cultures, and so they have something in them to either prevent it from being exposed to air, or to provide some sort of extra nutrients, or to prevent the organisms themselves from drying out. With your blood cultures, you see those two bottles at the bottom that actually look like bottles. Though your blood cultures typically have some sort of the SPS within the bottles that helps to preserve those organisms as well, and to help enhance their growth once it is loaded onto the analyzer. And then you can also have a heparin tube for, and it's not an actual, usually not an actual heparin tube, but it is a special tube for viral cultures, although you may not see viral cultures much within the lab. However, some of the things that should not be used would be EDTA tubes or ACD. And up in the top right hand corner, you can see an example of the ACD tube. And so something like this should not be confused with those urine collection tubes that are also yellow. So with these tubes, they are for HLA typing in blood bank or different types of DNA and genetic studies. So making sure that you use the proper tube is very important, and if you're ever in question, you always need to pull the specimen label back with the patient's name on it, just to make sure that the label on that tube is adequate for that particular specimen or culture that you are performing. Specimens that are not processed immediately can be stored, and dependent on the particular specimen and what organisms you're looking for will depend on the temperature. And so a lot of it will be dependent on if there's transport media involved as well. And so there is a chart in the book, table 5-1, that lists various specimens and what the storage requirement or the transport media that's acceptable for that particular specimen. And so, and it can be dependent too on exactly what organism you're looking for. And so depending on that, which you'll learn a little bit more detail about this once we actually start getting into those organisms, you will learn a little bit about which ones are more temperamental. And so depending on the specimen, it could be stored refrigerated, at room temperature, body temperature, or even frozen. So there are situations in which specimens would be rejected, although with micro, it is a little bit harder to recollect specimens. But with these situations when specimens need to be rejected, then that's something that may need to be discussed with the physician or the pathologist of the lab to determine if indeed you should proceed with that specimen. But typically the things for which a specimen would be rejected would be if it's mislabeled, if it has the wrong patient's name on it. So those would be rejected and need to be recollected. If it's not properly transported, so if it was stored at the wrong temperature, it was collected with the wrong swab, or if it was put in the wrong collection media, if it's been set in, got lost in the courier's car overnight and it's been sitting in the car in the hot for 24, 48 hours, then that specimen obviously couldn't be cultured, because those organisms would either have a lot of overgrowth, or depending on the temperature, it could have destroyed those organisms and it could have dried out. If there's not enough specimen to perform the culture, if the specimen has leaked inside of the bag, it could have been possibly contaminated. If it's in a fixative, of course this will vary by the specimen type, but some fixatives will destroy the bacteria. If it's an anaerobic culture that's requested, but the specimen that was sent, or the particular source that's listed on that culture, is known to have anaerobic organisms as the normal flora. And so an example of this would be a vaginal culture where there are anaerobic organisms that live within the genitalia vaginal tract. So ordering an anaerobic culture on that particular source means that you're going to recover normal anaerobic flora. And if you get the specimen and it's dried out, then obviously it's not going to grow anything if those organisms have dried out and died. Once the specimen is received into the lab, it should be documented with the date and time that it arrives. And so this is important, so you can compare with the collection date, with the received date, if there's any discrepancies in anything, and also to be able to track the specimen as far as when it was received and when it was actually cultured, when the results were sent out. So it kind of gives you a time frame on the specimen. Priority is always given to the most critical specimens. So for example, a spinal fluid is always considered a very critical specimen, and so those usually processed right away as soon as they come into the lab. You will also examine the specimen itself, and so when you do that, you may make a manual or an electronic record of that specimen as far as what that specimen looks like, or if there's any kind of issues with the specimen once it arrived to the lab. And when you perform the culture, you will want to make sure that you're looking for the bloody mucoid areas, depending on the specimen, to make sure with those mucoid areas or a pus type area of that specimen that you're most likely getting a bulk of those organisms. And so you do want to try to get those areas included as well. With a direct microscopic examination, you're preparing a slide directly from the specimen source. So for example, a sputum culture would come in a cup with the sputum actually in the cup. So you would make a slide off of that sputum sample. You would roll that onto a glass slide. And so what these type of examinations do, so rather than taking it directly from the culture and looking at the organism, you're looking directly of what come from the patient. You're being able to assess the quality of the specimen. So with sputum cultures, you're looking for a quantitation of white blood cells, epithelial cells, to see if it's possibly contaminated. It also will give an early indication of what can be going on with the patient. So for an example, with the spinal fluid, if you perform a direct examination of that spinal fluid and you see gram-positive cocci within that spinal fluid, then that gives the physician an early indication that there is some sort of gram-positive cocci organism that's growing within the patient, so they can go ahead and start treatment with antibiotics that target that particular organism. And you're also looking to see if what you saw on the actual gram stain matches with what you're actually getting in your culture. So if that spinal fluid showed gram-positive cocci, but when your culture grows you don't see any organisms, or let's say you see a gram-negative, then something somewhere in that process went wrong, so it needs to be investigated. So with direct microscopic examinations, there are several different staining techniques that can be performed. Some of these we've already covered in previous lessons, but the one that we're going to particularly pay attention now to is the gram staining technique. With your culture media, you have several types of culture media. And so they can be divided into categories. So you have a supportive media, which supports the growth of a wide range of microorganisms. And so this would be something like blood agar or chocolate agar. And you can see an example of that in the top left-hand corner, where you have a blood agar plate. You also have differential media. And so what this does is it helps to differentiate one organism from another just based on certain characteristics that that organism has. You also have selective media, which supports one particular group of organisms over another. So for example, it may support gram-negative organisms versus gram-positive organisms. And this is done by using some sort of dyes or antibiotics or other chemical compounds that inhibit the growth of the one organism but allows the others. And then sometimes you can have media that is both selective and differential. So for example, at the bottom left-hand corner, you see a McConkey plate. And so what this does is it allows the growth of gram-negative organisms. And it also is differential because it will differentiate the lactose fermenters from the non-lactose fermenters. So your lactose fermenters produce that pinkish purple color, whereas the non-lactose fermenters will stay kind of that clearish color. You can also have backup broths, which can be supplemental or enrichment. And so this is typically used in conjunction with your solid media, and it can help to detect when there's just a small number of organisms in that culture swab or whatever specimen has been submitted. Then it kind of helps to enhance that growth of those small organisms. And so sometimes something like this may be set up on like a vaginal culture would be an example, in which it would allow for an enhancement growth of those organisms, and then that broth itself can also be cultured or gram stained as well. So depending on which organism or pathogen you're looking for in a particular sample will depend on where you actually incubate those agar plates. So if you're looking for an aerobic type organism, these are incubated within an ambient air, and so this would go directly into the incubator with an oxygen of about 21% with a very low CO2. Your anaerobes cannot grow in the presence of O2, so those would go into some sort of like an anaerobic bag to prevent any sort of exposure to O2. Your capnophils require an increased CO2 with about a 15% O2 level. And then your microaerophils will grow under reduced O2 levels but increased CO2 levels. And so most of your medically relevant bacteria are facultative anaerobic bacteria, and so they can grow aerobic or anaerobic. Your temperature most of your relevant organisms will grow between 35, 37 degrees. And of course, with your funguses, we already covered that those have a pretty a lower range as far as where they grow at. So here we have a chart describing the oxygen requirements of the various organisms. And so with this, you can see a test tube with the agar media inside that tube. And so to inoculate these types of tubes, you would take a needle or the culture loop, and you would inoculate the top of that agar of that test tube, and then you would stab down into the bottom of the agar to make sure that that specimen passes not only the top but all the way through the agar to the bottom. And so with the culture being presented to the bottom of the agar, then you are allowing any sort of anaerobic growth to take place without the presence of oxygen. So with your aerobes, you have just growth where there's oxygen present, so closer to the top of that agar. And so when you have more oxygen kind of concentrating into that media, then you'll have growth around the top portion. The facultative anaerobes can grow in the aerobic and anaerobic conditions, so you will see it throughout the agar. However, you will have a little bit more of a growth at the presence of oxygen, so that would be along the top. With your anaerobes, those organisms would grow just at the bottom where there is no presence of oxygen. And then you have aerotolerant anaerobes, which typically just grow in anaerobic conditions, but they can grow in the presence of oxygen. So that growth will grow throughout that media because the oxygen really doesn't have much of an effect on it. And then you have your microaerophils. So with this, you only have aerobic growth. And so oxygen is required, but it's at a lower concentration. So you can see where it grows midway through the media. So it still needs that oxygen. It just doesn't need as much as your aerobes need. So with working up specimens or reading culture plates and identifying organisms within microbe, you can have limited identification in which you just kind of work it up to the genus but not the species, versus a complete identification where you're actually working it up to the point of narrowing it down to the particular species. With your presumptive identification, use a small batch of tests and presume an organism to be a particular organism based on those tests. So you're not going to do a complete workup, or it may be a presumptive identification for a preliminary workup before you do the additional automated type testing. With a definitive identification, you're also including the antimicrobial susceptibility testing. So typically this may be something that's going to be put on an analyzer, so not only does it have all the biochemical tests, it also has the antimicrobial sensitivity testing as well. You will also need to be able to differentiate the normal flora versus a pathogen, and so an example of this would be a stool culture in which you have normal microbiota within the intestinal tract, and so you need to be able to differentiate that which bacteria is normal versus something that may be a pathogen like Salmonella or Shigella. Critical values in micro includes bacteria growing in a blood culture, spinal fluid, finding something like a parasite, malaria in a blood smear, thick or thin, finding acid-fast bacteria, certain biological agents that you'll learn about in the future that are always considered a critical result, and some of those do have to be reported to the CDC. Anaerobes are considered a critical value. So with any of these, once it's found, it would need to be called and notified to the physician. So if you're doing a spinal fluid in the micro lab and you do a gram stain and you see organisms in that gram stain, then you need to call the physician and alert them versus reporting it in the computer. Something like a urine culture would just be reported in the computer and the physician would get the results and see the results versus having to call a urine result. Some terms that you should be familiar with are pathogens, which is a microorganism that causes an infection or disease. Virulence factors, these are characteristics that help the microorganisms to cause the particular disease. Pathogenicity and virulence is the degree to which the microorganism can cause a disease. Opportunistic pathogens or infections, these are infections that happen when one or more of the host defense mechanisms is disrupted, and then the infection is able to happen because the organism that typically resides in the individual is normal microbacteria, microbiome, and actually infect the patient at a site that is not supposed to be in. So here's some of the different virulence factors. Attachment is when the organism is able to outcompete colonizers and adhere to the surface. Invasion, usually by some sort of traumatic factor or due to the other virulence factors, the organism is able to disseminate into the deeper tissue. Survival against inflammation, and they do this by avoiding being phagocytized and by being recognized by the complement system. Survival against the immune system, or they can produce toxins. With endotoxins, it is actually in the cell wall of the usually gram-negative bacteria, and so once the organism dies, then those endotoxins are released from that organism to the surrounding tissue. With exotoxins, the organism, usually gram-positive bacteria, actually releases, it produces and releases that exotoxin. Some mediators of pathogenesis include a pathogenicity island or a biofilm formation. And so with your biofilm formation, there's an accumulation of the monomicrobic or polymicrobic population, so one organism or multiple organisms that will start to provide a protective environment for the organisms that live in that little cluster. So here you can see an example where number one, the organism attaches, and then from there it will start kind of building this little protective network, and the organisms will just continue to replicate and grow, and then as that protective network gets bigger, then those organisms can be released, and then they'll go and start their own new attachment and new colony. So the outcome of a patient with any sort of infectious organism will depend on a couple of different things. The state of the host's health, so how their immune system is, how it responds, their overall general health, the virulence of the particular pathogen that is the cause of the infection, whether or not the host can actually clear that pathogen. And so you do have three types of infection. You have an acute infection, which develops very quickly. Chronic infections develop slower, usually over an extended period of time. And you also have latent infections in which the organism can actually go into like a silent type phase where you don't have any sort of symptoms that present. Some of the signs and symptoms that can indicate an infection would include aches, pains, headaches, fever, swollen lymph nodes, rashes, any sort of sign of inflammation like redness, swelling, or heat at the site, coughing, sneezing, congestion, sore throat, nausea, vomiting, diarrhea. So a lot of these can indicate many illnesses, but it can also be a sign of an infection from a microorganism. So when we look into preventing infectious diseases, we look at a variety of things. So we look at different strategies to prevent transmission, to control the microbial reservoirs, and to minimize the risk before and after exposure. So some of these things could include just good hygiene, washing your hands, some of the things that happen within surgical operating rooms now as far as how they scrub in, how they scrub out, making sure that the instruments are cleaned properly. Some circumstances it may be that you're using disposable type materials now to prevent any kind of infection or hospital-acquired infections, and just things that you kind of use with your daily life of, preparing your food when you're cooking chicken, making sure that you prevent any kind of contamination off of that chicken, properly preparing your chicken so that you kill any kind of potential microorganisms that you could get through any sort of like foodborne illness. You can also use vaccines, immunizations to prevent infections, and so it can be active, which, this is typically what we think about for our immunizations, where it's something that's been modified and it's introduced into the body, and then there builds up a immune response. And so an example of this would be like a pneumococcal vaccine. And then you also have passive antibodies that are transferred temporarily. So this would be something like a mother passing to an infant, the antibodies that pass to an infant before the baby's born, and then the antibodies that are passed through breast milk. And with epidemiology, this is the science that characterizes the circumstances associated with how infectious diseases are acquired and how they are spread. So that's what the epidemiology field looks at, is the different infections and as well as ways to prevent those infections. So now we'll look at some of the different components of bacterial cells. And so with bacterial cells, they do have internal structures within the bacteria. Even though we can't really see those, they are there. And so here's just an example of the bacteria itself, where you have the wall, the outside cell wall, and then once you get internal into it, then you've got the different structures that are internal, and this can even be further broken down into those structures containing the DNA. So when we look at the outside of the cell or the cell envelope, we're looking at an outer membrane for the gram-negative, and then you have a cell wall. So the gram-positive has a very thick peptidoglycan layer, whereas the gram-negative has a thinner layer. So the gram-negative have that outer membrane as well as that thin layer. And then your acid-fast bacteria have a waxy substance that is on the outside of that bacteria. Then you have the perioplasma or the perioplasmic space, and this was within your gram-negative. And so this contains a gel-like substance that helps to capture nutrients, and it also contains enzymes. And then you have the cell membrane or the cytoplasmic membrane. And so this is the deeper layer of the cell, and it contains proteins and enzymes that are vital to cell metabolism, and it also serves as an osmotic barrier. And so here you have an image with that gram-positive versus the gram-negative, and so you can see that peptidoglycan layer for your gram-positive, whereas your gram-negative, it has a very thin layer, but then you also have that outer cell wall. And so some of your organisms can actually have a capsule on the outside of that cell, that bacterial cell. Some of them are able to form slime type layers, and then some of them can also create those biofilms. And we saw that image on a couple of slides back where you can have one particular organism or several organisms that create that biofilm. Some organisms actually have some sort of appendage to it, so it may have flagella or phyla that help it with attaching. So with the capsule, that exterior layer that protects the bacteria, it actually helps it from being attacked by the immune system, and it also helps to make those biofilms. And then with phyla, you have the hair-like projections around the outside surface, and so it helps with the attachment of that organism. And then some of them also have motility, so they're able to move with the flagella. And so depending on the location of the flagella, it actually can have a term with that placement. So if it's polar, it just has one single flagella, whereas the more flagella that it has, or depending on the location of it, it changes that term. So with a gram stain, this is the most used stain within the laboratory for viewing and differentiating bacteria. And so what happens with this is that when you use the stains with it, so with your crystal violet stain, it forms a complex with the iodine, and so this will be present in your gram-positive organisms, whereas your gram-negative organisms do not have that thick peptidoglycan layer, so they are not able to retain that iodine complex to stain that purple color. So with this, they would be gram-negative and they would be pink. And so what you have is a primary stain, which is your crystal violet, a mordant, which is your grams iodine. Then you use a decolorizer, and then you use a counter stain, which is a saffron, which is a real pretty pink color. And so your results would be a gram-positive, which stains the deep shade of purple, and you can see that image here, or gram-negative, which stains pink. So you should be familiar with this procedure. You will not have the opportunity to perform a gram stain this semester, but when you do your fall semester class, you will do a gram stain. And so what happens with the gram stain procedure, you will prepare a slide with your organism straight off of the plate or with a direct culture, so from a sputum culture or swab. And so once you have put that sample onto the slide, you allow it to dry, and then you will fix that material to the slide either by a heat fix type method or with something like methanol. Then to actually stain your slide, once your slide has cooled off, if you've heat fixed it, then you will flood the slide with crystal violet, and you'll let it set on the slide for a few seconds to a minute. It will depend on the manufacturer of the reagent as to what the timing is. But once it has set, then you will rinse that off with water, and then you will flood the slide again with the iodine, and this allows that crystal violet iodine complex to take place. And then after you've let that sit for the allotted amount of time, then you will rinse it off. And then from there you will use the decolorizer. And so with the decolorizer, you don't want to over decolorize and remove too much of that dye, but you also want to be sure to remove the dye from your gram-negative bacteria to allow them to absorb that saffron. And so by doing this step, you will run the decolorizer over the slide, and you will watch it run off the slide until it no longer produces any of that purplish blue dye. And so once that runs clear, then you will rinse that slide off, and then you will flood it with the counter stain or the saffron, and let that sit, and then you will rinse that off and let it either air dry, or you will press it between paper to dry off any of that moisture. And then after it is dried, you will look at it on the 100x objective, or that would be 1000 magnification, and this will be your oil objective. And you're looking for any sort of white blood cells, red blood cells, bacteria, or anything else that may be present. And so here again, it just goes through the process that's taking place with the crystal violet. Everything is stained purple. And then once you add the iodine, you form that crystal violet iodine complex, and it will stay within the gram positive, but once you decolorize it, it will be removed from the gram negative. And then your saffron is your counter stain, and so that will stain your gram negative that pink color. If you over decolorize the specimen, you've let that decolorizer run on that sample too long, so when you're running it over and you see that purple color come out and it kind of stains that decolorizer a purplish bluish tint, if you keep putting that decolorizer on it after you see that it has started to run clear, then you'll actually start to remove the color from the gram positive, which could in turn, they would get stained and they would appear gram negative. With your gram variable bacteria, you have intermittent type staining, and so this would be where you have gram positive and gram negative, and this can be due to the particular organism, where maybe it's got some sort of capsule or something around that cell, or it could be something like the patient's been treated with antibiotics, so the organism could be dying. It could be the age of the culture. So there are a lot of things that can cause that gram variability. And so what that would look like would be something like in these two images, where you've kind of got a little bit of that purple color, but you've also got the dark red color rather than pink. And so they get a different kind of shade to them. And so this is where your training and recognizing what the bacteria look like will help you to be able to detect when something like this has happened. So not only do we look at how it stains as far as gram positive or gram negative, but we also look at the shape and arrangement of the bacteria. So they are very small bacteria, but they can come in different shapes. So you have cocci, which are circular looking organisms, coccobacilli, which are bacilli that are kind of oval shaped, so they're a little elongated. You have bacilli, some people also refer to these as rods, so they're more of a rod shape. A fusiform, which is pointed on the ends. You can have them have a bit of a curved shape to them, or they can have a spiral shape to them. They can also be arranged in singles, pairs, groups, so you could have tetrads. So if you look at the image here, you can see some of your cocci where you've got just a single one versus a diplococci, which are two. Sometimes they can be encapsulated. You can have them in groups or clusters, so you can see a group with the staphylococci, tetrads in groups of 4, and then you can also have them in chains. And so here you see the streptococci that are in chains. With your bacilli, they also can be in the same formation or similar formation. And then you can also see on the side where you have your fusiform type bacteria, where it's kind of got pointed ends. And then at the bottom you can see your spirochetes or your corkscrew formed of bacteria.